So my name is Sylvia Boj. I'm the Chief Research Officer at Hubrecht Organoid Technology, and I'm responsible of the research team and the research activities that take place at HUB. By predictive diagnostic or personalized medicine, we mean the capacity to identify the right treatment for each individual patient. Using as an example oncology patients, we are aware that there are different treatments to treat this type of patients, but not all the patients have the same response. So with HUB Organoid Technology, we aim to generate organoids from the tumors of these patients, expose them to the drugs available, and identify the right treatment for each individual patient. We are aware that drugs and treatments generated in the past were not necessarily having the same impact in all patients, and that there is a need to identify the right treatment for each individual patient. And we believe that with organoids we can provide an in vitro test. We can bring the patient in the lab and test, for example, in oncology, the treatments available on first-line treatment, and identify what will be the group of drugs that will benefit or will have an impact in the patient. Despite that we will define organoid technology a very easy technology to implement in any lab, at the same time it's a complex model, or I will say a sensitive model, more close to primary tissues. So having high quality products ensures the success of establishing this technology in the lab. So very good products from Corning like Matrigel are essential for the success of establishing these models. Actually, we have a strategic collaboration with Corning to validate some of the reagents as organoid-proof reagents. So if I have to provide a tip to get the best cell culture results, I'll suggest first following the standard protocols that has been developed for establishing organoid technology, and also using high quality reagents to ensure a good expansion of the models. But a tip that may be more close to people working in the lab every day with organoids: when manipulate them, try not to disrupt them to single cell. They recover better when they are disrupted as fragments of multiple cells, not single cells. And we've seen also an improvement if during the manipulation for disruption, the organoids are not kept on ice, but just manipulated at room temperature. So one of the challenges that we encounter for the technology, since our aim is to make it available for the community, is to educate about the complexity and certain requirements that working with this technology are needed to succeed on the establishment. Just to bring an example, when working with tumor models, meaning when organoids have been generated from a tumor, from a patient with colorectal cancer or breast cancer, with our technology, we are able to maintain the heterogeneity of the tumor, but if this organoid culture is not cultured properly, it will be in a matter of weeks or months that this culture does not represent the original heterogeneity, and that then results in terms of gene expression or drug tests will differ, not because of the robustness of the model, but because the model has not been manipulated properly. In terms of opportunities, we want to extend the success that organoids or HUB organoids have meant for CF community to other monogenic diseases. So for cystic fibrosis, we've been able to generate organoid models that can predict patient response, and that have been already able to provide access to patients to treatments that in any other way they will have not been able to get the access. So we are looking for similar genetic diseases in other system models like liver organoids, where we can have this impact in the patients.